Our team Bio-Spire is aiming to develop an efficient and quick way of detecting sepsis. Every year, approximately 1.7 million American adults develop sepsis, and nearly 300,000 people die from it in the US alone. In fact, 1/3 of all patients who die in American hospitals have sepsis. In 2013, the United States spent around $24 billion in treating sepsis. The sepsis is not only a problem in the US, however. 85% of sepsis cases worldwide occur in low to mid-income countries. Furthermore, 20% of all deaths in the world are caused by sepsis. Sepsis can initiate from a small infected wound or post-surgical complications and is characterized by its development into an overly strong immune response, leading to very low blood pressure and life-threatening organ failure. According to the policy currently employed by the Critical Care Unit in the University of Rochester Medical Center, medical professionals should aim to start treatment of sepsis within the golden hour of sepsis diagnosis, which emphasizes that with every hour that sepsis treatment is delayed, chances of survival decrease significantly. Currently, the standard detection method to diagnose sepsis is to culture a patient's blood and check for pathogens that cause the infection. Unfortunately, this method takes 24 hours to produce results and can yield false positives, and also requires expensive equipment and reagents. This is where we want to make a difference, create a faster, non-invasive device to continuously monitor a patient at risk of developing sepsis. During an infection, the body releases biomarkers, which are molecules associated with a particular disease. Our device will be used to track increases in biomarker concentrations associated with inflammation and other symptoms of sepsis. While many of these markers stay in patients' bloodstream, some are exported into their sweat, and those can be detected and quantified by our device. Unlike blood, the detection of biomarkers in sweat is non-invasive, and our modeling has demonstrated that levels of biomarkers in blood and sweat are typically correlated, such that changes in the concentration of sweat biomarkers are indicative of changes in biomarkers in the blood. Our software thus correlates the biomarker values in sweat to those found in blood to provide more familiar data to medical professionals who are used to interpreting blood biomarker data. Since sweat can be continuously drawn and analyzed, it provides medical professionals with continuous quantitative data on patient biomarker concentrations that deviate from the healthy range, and eliminates the need for constant blood draws and lengthy waiting times for test results. Upon visiting the intensive care unit, we learned that the best location for our device is on the forearm, since that is the location that was available in majority of patients in the ICU. After talking to the Director of Adult Critical Care Nursing, Kate Balson, and the Director of Critical Care, Dr. Pietropoli, from the University of Rochester Medical Center, we learned that the postoperative patients are the best target for our device. This is because we would be able to monitor the condition of patients as soon as they're out of the operational room, having a very concrete start time of our data. For this reason, we would be able to not only track the changes in the levels of biomarkers, but also compare them to the standard biomarker levels for a particular patient. When designing our apta sensor, we realized that we would be dealing with very small amounts of sweat due to sedentary ICU patients. As a result, we developed an apta sensor that incorporates graphene screen-printed electrodes and microfluidics in one device. Microfluidics is the control of very small volumes of fluid, and we incorporated this to collect, handle, and transport sweat to electrodes in our device. Our microfluidic device is composed of 3 main sections: capillaries to collect sweat, a long white channel for electrochemical reactions and outputs, and a final micropillar reservoir for used and excess sweat. The white channel in the middle contains electrodes modified with reduced graphene oxide, which allows biomarkers to bind and create current change, and allows us to measure impedance through resistance. Using our software, we will display these changes to the medical staff in hospitals.
After consulting with experts Jeffrey Beard, a PhD student at URMC working on microfluidics and cost-effective biosensors, and Dr. James McGrath, a professor of our microfluidics course at the University of Rochester, we decided to use an inverted pine tree-like structure composed of narrow capillaries for the collection of sweat. This part is in contact with the skin and allows the sweat to wick into the capillaries. Due to pressure differences, sweat moves slowly towards the bigger capillaries and the electrodes. A single closed channel that is long and wide will be used for the electrochemical readout. To get an accurate readout with the smallest amount of sweat, we put the electrodes in series. Sweat will flow to the final micropillar reservoir and evaporate after flowing over the electrodes. This micropillar also acts as a passive pump to wick the sweat all the way to the end of the microfluidic device. The patients that we were targeting are not likely to sweat a lot. In order to have sufficient sweat volume to flow through our microfluidic device, sweat has to be locally induced using pilocarpine iontophoresis. Since we didn't have access to patients, we proposed this as a future direction for our sleeve. We chose to 3D print our microfluidic device, not only because it is cheaper and easier to use and manipulate, but also because it's better than the traditional way to manufacture microfluidics, which is to use photolithography, requiring more expensive equipment and a clean room. We used the simulation software COMSOL to make an initial design of our device, and we estimated what flow rate might be optimal for biomarker binding. Then we designed a 3D microfluidic device in Onshape, a design software, printed the mold and used it to make our microfluidic device by pouring a flexible material into it. We originally tried to use a heat-shrinkable material for our device, but the stiffness of the cured product wouldn't allow fluids to flow through it. So instead, we decreased the size of our 3D printed mold and directly poured PDMS into the mold to make our device. To test the fluid flow through the device, we used diluted food colorings. At an injection rate of 0.1 milliliters per second, the velocities we measured were 6.3 millimeters per second for sweat collection for the capillaries, 8.1 millimeters per second on electrode channels, and 7.2 millimeters per second for the micropillow section. In the next experiment, we injected food coloring at different rates and showed that the velocities in the channel decreased as the injection rate decreased. This experiment led us to the question, how do we determine the optimal sweat flow velocity over the electrodes that still allows the biomarkers to bind to the aptamers, provide an electrical readout from electrodes, and then dissociate? To answer this, our modeling team used kinetics equations for binding and Navier-Stokes for fluid flow in MATLAB to determine the sweat flow in the microfluidic device. We found that the inlet flow rate necessary was 130 millimeters per second. The hardware team used this value and found that the channel dimensions of our device sufficed to obtain this inlet flow.
Next, what are aptamers and why did we decide to make an aptasensor? We identified 4 biomarkers that had multiple reported clinical values for sepsis: CRP, TNF-alpha, IL-1 beta, and IL-6. Researchers suggest that CRP in combination with IL-6 is very effective for detection, and CRP levels are expected to decrease in post-operational patients unless there's an infection present. Among these biomarkers, TNF-alpha, IL-1 beta, and IL-6 are all pro-inflammatory cytokines that are quite common in other diseases. CRP is secreted by the liver in response to high presence of IL-6 and other cytokines, and it is part of the innate immune response. To increase our specificity for sepsis detection, we also picked lactoferrin, a protein involved in innate defense against microbial growth, which is also found to predict possible multi-organ failure during intestinal inflammation. Medical professionals and researchers such as Dr. David Nagel at the University of Rochester Medical Center approved the feasibility of using these 5 biomarkers together because they are both present in sweat and efficient in sepsis detection. The next step was to find biological parts that can detect these biomarkers even at very low concentrations. We decided to use an aptamer-based diagnostic device which uses nucleic acid aptamers as the biological sensor. Our nucleic acid aptamers were single-stranded DNA that adopt a tertiary structure able to specifically bind a target molecule, specifically the 5 biomarkers we were interested in. We selected sequences that demonstrated a high affinity in literature and synthesized them as biobricks. Our aptasensor requires the use of high concentration of these single-stranded DNA aptamers, and because they are single-stranded molecules, conventional PCR is not an option. To make our aptamers, we utilize asymmetric PCR, which is similar to normal PCR, except the forward primer is in excess of the reverse primer. As the limited reverse primer gets consumed after the first few cycles, the reaction shifts forward toward single-stranded DNA production in a linear manner. This occurs until the increase in the number of DNA copies is restricted by the amount of enzyme present.
Once we had produced aptamers that could bind to our biomarkers, we needed to find a way to detect when this binding occurred. We decided to attach the aptamers to reduced graphene oxide, or RGO, because this would allow us to detect the binding through an electrical signal sent through the highly conductive graphene sheet. Reduced graphene oxide is a 2-dimensional sheet of 6-membered carbon rings. The sheets are decorated with few oxygen-containing groups like hydroxyls, epoxides, and carboxylic acids. Graphene oxide, or GO, has a similar structure but with more oxygen groups. When GO is reduced, the oxygen groups are replaced with carbon-carbon double bonds, eventually forming an extensive system of conjugated p orbitals, also known as a pi system. This makes RGO much more conductive than GO, since electrons can flow through the conjugated pi systems. When we were deciding on a material for an aptasensor, we needed one that was conductive enough for us to be able to monitor small changes, inexpensive enough to be widely available in hospitals, and easy to produce. RGO meets all of these criteria. After discussing with Dr. Benjamin Lena from TU Delft, Netherlands, we learned that there are 3 ways to reduce graphene oxide to RGO: thermally, chemically, or biologically using bacteria. Thermal reduction of GO requires high temperatures, which could cause the double bonds in RGO to interact with one another and cause defects in the structure of our RGO sheets. Chemical reducing agents are either too hazardous or too strong, so that they remove too many functional groups that we need to attach our aptamers. The third method, bacterial reduction of GO, fits our project the best because it can be done at room temperature and without hazardous reagents. We use the bacteria Shigionella oneidensis MR1, as it has the capability of reducing insoluble compounds like GO. Although bacterial reduction seems better than the other 2 choices, this takes more time than the chemical reduction of GO. Ultimately, we still decided to use bacterial reduction because it's safer, more accessible, and more effective for our project than any other reduction method. Shionella oneidensis MR1 reduces graphene oxide via extracellular electron transport, which may occur in one of two ways. 1. The flavin mononucleotide pathway, which works via electron shuttles, or through membrane cytochromes, which work by electron transport. Flavins are moderately strong reducing agents that can leave enough oxygen groups on the RGO to which we can attach our aptamers. Since reduction by Shionella can take longer, we aim to speed this up by overexpressing genes involved in the reduction process. We look specifically for genes to modify and express in Shionella that either increase the amount of electrons that Shionella could export via membrane proteins or increase the amount of electron shuttles. To determine which genes to modify, we built a partial metabolic model of Shionella oneidensis using the software Cell Collective, which informed us that changing gene expression would definitely give us increased reduction ability. Our model showed us that theoretically, overexpression of multiple genes in the flavin pathway, as opposed to just one, could be more beneficial to increasing the speed of reduction. Wet lab successfully showed that overexpressing rib cluster genes wouldn't be detrimental to Shionella since they survived and were able to reduce GEO. With this modeling, we decided to overexpress these genes to increase the rate of reduction. Overexpression of these genes should lead to more direct contact of the outer membrane cytochrome proteins with graphene oxide, which should increase the rate of reduction. The rib cluster, RIBF and OPRF, are involved in the production of transport of electron shuttles for electron transfer during reduction. The rib cluster is a group of genes that are a part of the riboflavin synthesis pathway. Overall, the overexpression of these genes should increase the amount of electron shuttle flavins that the Shionella produces and should increase their export into the surrounding media, which will lead to an increased rate of reduction of GEO. OD at 600 nanometer measurements were taken hourly over a 48-hour period for microbial reduction. We saw that with a 5-hour induction with 1.5 millimolar IPTG prior to reduction, YDEH has the fastest rate of reduction. This measurement shows the maximum rates of reduction are seen to be greater than wild type for rib cluster, RIBF, OPRF, and YDEH under different induction conditions. Raman spectroscopy showed that after 48 hours, all the transformed strains had similar levels of reduction. However, spectra of RGO after 12 hours of microbial reduction showed that SIME-A and rib cluster had greater levels of reduction than wild type.
To attach our single-stranded DNA aptamers to RGO, we chose to use a cost-efficient method that does not require any aptamer modifications, pi-pi stacking. Pi-pi stacking allows for aptamers to attach to RGO simply via non-covalent interactions between double bonds. Our discussions with several professionals working with aptamer-based biomaterials, such as Dr. Benjamin Lerner, Dr. Richard Yannison, and Dr. Cecilia de Carvalho Castro at Silva, confirmed pi-pi stacking seems feasible and that the biomarker binding should result in detectable electrical change on RGO. For the purpose of our aptasensor, we wanted to know what density of functional groups we should aim to have on the RGO. Functional groups are mainly oxygen-based groups attached to the graphene oxide that decrease its conductive properties, so we want to avoid having too many of them. However, they are necessary to make the material hydrophilic and thus interact optimally with sweat. The question becomes, what concentration of aptamers should we use to obtain optimal aptamer attachment through pi-pi stacking? To answer this, we used data reporting RGO with various degrees of reduction, where the material was characterized, then produced scatter plots and determined the correlation between oxygen-containing functional groups and the number of pi bonds. By introducing the Raman spectroscopy data from wet lab to this model, we determined the specific surface area of the produced RGO. Then we calculated the optimal concentration of aptamer. With the optimal aptamer concentration, surface area, and pi-pi stacking method, we successfully attached aptamers to RGO.
Once biomarkers bind to the aptamers that are attached to the RGO on the electrode, there will be a detectable change in impedance, which will be quantified and used to calculate the concentrations of the biomarkers. Each biomarker sweat patch will be connected to an individual circuit for signal amplification and readout. Impedance is the opposition to alternating circuits presented by the combined effect of resistance and reactance in a circuit. To measure impedance changes once the biomarkers bind to the aptamer, we manufactured a low-cost potentiostat. Our potentiostat was made to measure changes in direct current, which could be used to calculate resistance using Ohm's law. Although impedance is effective resistance to alternating current, since resistance is the real part of a measurable quantity of impedance, our potentiostat was able to detect electric changes once the biomarkers bind. By keeping the potential constant, the change in current was recorded as the biomarkers were binding aptamers. To detect binding of biomarkers to the aptamers on the RGO sheet, we drop-casted aptamer and RGO onto low-cost screen-printed electrodes for microvolume samples, and then measured the current change using our potentiostat. As these electrodes are small and thin, they have minimal impact on the parameters of our microfluidic device. The working electrode is modified with a graphene solution by using a thin needle to add GEO on top of it. Then a PDMS electrode cover that we designed on Onshape keeps the electrode bound to the microfluidic device. To relate the resistance to the concentration, we exposed RGO sheets with bound aptamers to sweat mimic solutions containing various known concentrations of each biomarker and measured the resulting resistance. This helped us experimentally determine a mathematical relationship between resistance and concentration, to which we can plug a value of resistance into and get our concentration value. To show that our aptasensor would detect the presence of a biomarker in sweat, we prepared concentrations of lactoferrin biomarker from 1000 nanomolars to 1.9 nanomolars in sweat mimicking solution with a pH of 6. We modified the working electrodes with either a solution of RGO and 10 nanomolars of aptamer, or just with RGO, which was our control. Additionally, we had a second control where we transferred only sweat at either pH of 6 or pH of 4.7 without any biomarkers on the top of the electrodes modified with either the aptamer and RGO or just RGO. We used our potentiostat to conduct cyclic voltammetry to detect electrical changes once the biomarker binds to the aptamers. When lactoferrin aptamer was attached to RGO, we observed an increase in resistance with increasing biomarker concentration until about 400 nanomolars. The data increased monotonically in a range from 50 nanomolars until around 450 nanomolars. Afterwards, the resistance decreased again, which could be explained by aptamers being saturated with biomarkers. These findings led us to believe that our potentiostat can successfully measure biomarker concentrations until saturations of aptamers is reached. For medical professionals who diagnose sepsis, who are familiar with blood concentration values, we converted the predicted sweat concentration into a blood concentration using another linear regression analysis based on published data to estimate the line of best fit for each biomarker using Python. Our software, which is designed to be easily adapted to diseases other than sepsis, takes the resistance values from our potentiostat and uses the equations determined by our models to convert the resistance data into sweat biomarker concentration, then to blood concentration, and finally shows a yes or no reading on whether or not the patient has sepsis based on each biomarker. Our team has extensively studied the cost of production, details of use in clinical settings, and commercial implications of our device. In order to gauge the market need, we tested product market fit and conducted customer research to make sure our device is novel. We met with numerous healthcare professionals and FDA employees in order to make sure that our device is following guidelines for medical aptasensors. The professionals we talked to in URMC view the biomarker approach as the future of sepsis diagnostics and have assured us that their hospitals would have a strong interest in purchasing this type of device in order to improve the quantitative diagnosis of sepsis. Furthermore, we determined the cost of production would be cheaper than most aptasensor products.
Our outreach activities reached over 380 students, from elementary-aged Girl Scouts to incarcerated individuals, and all the way up to senior citizens living in retirement communities. We created a series of different synthetic biology activities to share with our students, including microfluidics, pH, DNA extraction, genetics mystery, and design your own plasmid activities. Every time we ran the activity, we collected feedback from our participants and incorporated this feedback into improve our activities. For example, we received feedback from students to include visuals in our pH experiment. So we created visual guides and used them in the following times that we did the experiment. We really enjoyed being able to share information about synthetic biology with students and inspire them to learn more about science and how it can be used to improve the world for the better.
Throughout the past months, we've made great progress and were able to build a prototype encompassing the most important components of what we envision our device to be. We were able to produce RGO and drop cast it onto our screen-printed electrodes and were then able to attach our sepsis biomarker-specific aptamers to the RGO to measure the presence of our biomarkers with our self-built potentiostat. We optimized our microfluidic device to be able to collect sweat and transport it at the required velocities to our electrodes, and finally assembled all individual components together into a sleeve-like device that can be worn on the forearm and can be adjusted to many sizes. Our novel device has the potential to revolutionize the field of medical biosensors, as it can be easily adapted to any other medical condition. In the future, we could thus not only be able to drastically reduce fatalities due to sepsis, but could also detect a variety of different illnesses and save millions of lives. Thank you for your time.